our topic today is basic plant pathology. I know as we've gone through several of the classes, I know with the turf grass class, they've talked some about diseases of turf grass. When Michael Easley was here talking about trees, he talked about some of the diseases affecting trees. I don't know if Cheryl talked any about rose diseases last week or not. But you know, all of our vegetables, our roses, our landscape plants, they're all susceptible to disease. And you know, it's a little more challenging when we're talking about diseases of plants because unlike people or children or pets, you know, they can tell you, I hurt here or I don't feel good, or maybe our dogs or cats might not be able to say ruff, 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 and we know what that means, but you know, we can kind of tell by their actions that something's not right. Well, with plants, it's a little more challenging and difficult to tell when something is wrong because we don't see or realize something is wrong until they're showing those symptoms. And by the time they're showing those symptoms, in some cases it might be too late to do something to effectively treat them. So by knowing some of the things that can affect plants and maybe some of the signs or symptoms to look for, maybe you can just have that knowledge base built up so that you can know what to do and maybe even keep those plant diseases from spreading to other plants. So we're going to start off with just some central disease concepts. We're going to talk about what causes plant diseases. We're going to move into, okay, maybe how do I diagnose this? What's going on? What do I do? Where can I send this for positive diagnosis? And then I'll pass along some resources for you as well. All right, well, we're going to get started with basic plant pathology. So when we're talking about plant pathology, diagnosing or realizing what causes those diseases within plants. Well, first, I guess we need to define the term plant disease. What is a plant disease? And a plant disease is any continuous irritation. Now, for us, an irritation, like if it's a scratch or an itch, not necessarily so for a plant, it's just an irritation or maybe some sort of abnormality caused by a biological pathogen or an environmental pathogen. So we're going to talk about some of those biological pathogens, fungi, bacteria, viruses, nematodes, but that's not the only thing that causes disease. It can be caused by some environmental factors as well. Maybe the pH of the soil, maybe drought, maybe too much rain. We're going to talk about some of those environmental conditions. So it's a continuous irritation caused by either a biological or an environmental pathogen that hinders normal plant growth. So it alters that plant in some way so that it doesn't grow normally. And it could be something such as a bacterial spot on this pepper here. We might see these odd types of growth coming out of maybe the plants or the plant stem. Maybe it's a fungal issue. Or with these pumpkins here, you can kind of see that those are deformed. Those are a little deformed. They're not the varieties of pumpkins that have the warts and stuff on them. That just doesn't look good. And in that case, those pumpkins are affected by a mosaic virus. So all of these pictures here depict a biological pathogen. But it's hindered normal plant growth. It doesn't look like what they should look like. And these plant diseases, there's several effects that it can have. It can, of course, damage, when we're talking about a plant disease, it can damage the whole plant. It can damage the stem, the roots, the leaves. It can damage all of those plant parts. Or you might just see that plant disease at maybe just a certain stage of plant growth. You might see it during all stages of plant growth. So, these plant diseases can certainly have an impact on the parts of the plant that they damage. And these plant diseases, they can reduce the quality. We go out to our vegetable garden and we want to have those nice big juicy red tomatoes. Well, if we've got blossom end rot, or if we've got insect damage, or if we've got something that's affected the quality of the tomato, well, if it's from our garden, we might go ahead and eat it, but would we purchase that product at the grocery store? Probably not. So these diseases affect the quality. They also can affect the quantity that's produced. So if you go out to your garden and your tomatoes have some sort of plant disease, they're not going to be as vigorous. They're not going to be as productive. You're not going to get as many tomatoes as you would if you had a nice healthy plant. So These diseases affect the quality of the produce. They affect the quantity. Raises the production cost. We go to the store and we go to buy produce and we're scratching our head thinking, why in the world is this carrots, these vegetables, these broccoli, why do the vegetables cost so much? Because the producers are having to treat and care for in many cases, preventative measures for diseases that adds to the production cost. And because we have come to expect a relatively inexpensive food product, we want it at the cheapest price. We're not going to buy it if it's a higher price. Well, the farmer has to cover their cost. You as the producer want to cover your cost. And if we're not going to pay those big prices, ultimately at the end it reduces profits, either from the producer or from you. Because if you're having to add all of these extra inputs in order to produce your vegetable garden, and those profits aren't necessarily economic profits. It could be, just, I got to go treat the garden. I'm having to pull up these tomatoes and these vegetable plants. Some of the profits could be those intrinsic or those interior rewards that you get as well. So a lot of effects from plant disease. Now, what causes plant disease? In order for a plant disease to occur, in order for disease to occur, not necessarily plants, but even in humans and animals, three things have to be, the stars have to line up with those three things, and we can look at that kind of in like a triangle. And we refer to that as the disease triangle. So in order for disease to occur, we have to have a host. And that host is going to be our crop or that variety of plant that we're growing. We need to have a pathogen, and that virulent pathogen or that pathogen that's going to cause a disease can either be a fungi or a bacteria. It could be a virus or a nematode. Now, we don't hear a lot about these, but I threw them in. Mycoplasmas and spiroplasmas. They're actually forms of bacteria, but whereas with fungi and bacteria, they have a cell wall that make up the organism. These mycoplasmas and spiroplasmas are actually forms of bacteria that lack a cell wall. So very, very primitive in nature. We don't hear a lot about these. A lot of times they're lumped in with bacteria. So we need to have a host, we need to have a pathogen, and then the environment needs to be right in order for that pathogen to successfully infect the host. And for the environment, it might be the air temperature. We tend to see plant diseases more in the summer months whenever the temperature is warm, because a lot of these fungi and bacteria can't survive in the cooler temperatures. So maybe the air temperature needs to be a certain temperature, or even soil temperature. We've got bacteria and fungi that might live in the soil. So maybe the soil temperature needs to be above a certain, maybe it needs to be above 70 degrees in order for that environment to be just right. Soil fertility. If we have good soil fertility, many times our plants are going to be happy and healthy. But once we let that soil fertility get out of balance, maybe we don't have the nutrients needed to have a healthy plant, we're going to weaken the immune system of that plant. Maybe we don't have enough nitrogen, for example. And so we weaken the immune system of that plant. It makes that plant more susceptible to a disease. Soil type can even affect whether or not a pathogen can affect a plant. Soil pH, rainfall. Maybe it can be too much rainfall. We've got a lot of rainfall and we've got a high relative humidity. Maybe it creates a moist environment. We're going to see maybe an increase in fungal growth. Or maybe we don't have enough rainfall and we end up with some drought conditions. And when we have something that might look like a plant disease, but maybe it's just the plant suffering from drought. So, and then of course soil moisture. So in order to have a disease, all three of these must be in balance: the host, the pathogen, and the environment. So we can easily minimize our risk of plant disease by getting rid of one of these. So if we have a host, I'm just going to a plant, and we can maybe plant a disease-resistant variety, and that will and that's getting rid of one of the parts of this soil triangle. But let's say we've got a variety, and we know, well, I really like this variety. It was passed down from my grandmother, and it's been in the family for a long time. I really like it, but I do know it's disease resistant. By altering the environment, by removing some of those conditions from the environment, we can reduce our risk of getting a plant disease. So for plant diseases, we got to host the pathogen in the environment. All three of those, like that, those stars have to line up in order for that disease to occur. Unfortunately, though, more times than not, we have a lot of instances where those stars line up and we get plant diseases. But by removing one of those from the triangle, we've reduced or greatly reduced our risk of getting a plant disease. So, a susceptible host. That susceptible host, that plant, provides food and water or shelter for another organism. We might not think about fungi or bacteria seeking shelter, but the plants that they live on, that's their home. Whenever Michael Easley was here talking about trees, he talked some about hypoxylin fungi. Hypoxylin is a fungal problem, and he said all of our trees have hypoxylin fungi living in them. It's just when that tree becomes stressed, does that fungus really begin to grow and develop and take over the tree. So in that case, our susceptible host is the tree. Now the environment that includes temperature, our moisture level, our soil structure, the wind, light conditions, herbicides, and then our pathogen, the viruses, the bacteria, fungi, nematodes. So with our hypoxylin canker, the susceptible host was our trees. The pathogen is that hypoxylin fungi. Whenever that tree becomes stressed, usually because of a lack of moisture, because our trees become stressed during drought condition, that's when all of the conditions are right for that tree to become infected by the hypoxylin. So in that case, in order to keep the hypoxylin from taking out the tree, we can alter the environment by making sure that tree gets plenty of water during those drought periods. The tree stays out of stress, and so that hypoxylin fungus is going to stay at bay. So by simply altering the environment, we can help control some of our plant diseases. So we talked, what can make a plant sick? We've talked about viruses a little bit, and bacteria, fungi. I haven't mentioned parasitic plants yet, but something such as mistletoe, something like dodder. We'll talk about dodder here in a little bit. Nematodes, insects. Insects and mites can transmit diseases to plants. So these insects and mites, they'll feed on our plants. They can transmit bacteria or even viruses to plants. Other herbivores, other plant-eating animals feeding on our plants. These are all living or biotic means of disease transmission. Now, whenever we look at disease, really it's the viruses, the bacteria, and the fungi, the parasitic plants, nematodes to a certain extent that cause disease. Insects, other herbivores, they cause more injury that could lead to disease. But then we also have these non-living factors that cause diseases. Air pollutants, other chemicals, maybe herbicide, the soil acidity or alkalinity, nutrient imbalance in the soil. Maybe we've got low oxygen content, which maybe could be as a result of air pollution, drought, too hot, too cold. We go out there and we, with our weed eater, and we maybe cut up or mar the bark of the tree. We've got a mechanical impact. So these injuries can lead to disease as well, because you've got an open wound. You've got a place for that fungi or that bacteria to enter into that plant. So there's a lot of factors that can make plants sick. And I failed to mention, genetic abnormalities. That's another avenue that plants can get sick. So it's not just fungi, bacteria, viruses, and nematodes that make plants sick. Sometimes just injuring the tree or having those environmental conditions weaken the tree or the plant such that it makes it more susceptible to those plant diseases. So as we talk about plant diseases, there's kind of 6 steps that we can follow or that we can outline to how that plant disease spreads or how it gets established and spreads. And the first step is that pathogen is deposited on the plant. Either blown through the wind or transmitted by an insect, it's deposited on the plant. After entering that host, after entering that plant, the pathogen begins parasitizing it, basically living off that plant, living off that host. So it penetrates, gets into the plant, and starts parasitizing it. And then that leads to an infection. That's the nutritional relationship between the pathogen and the host. And that's when that infection takes place, that's when we're really going to start seeing some of those signs and symptoms that the plant is being affected by something. From that infection, we're going to see colonization. The plant is going to move into other parts of the plant. So we might see it starting in a leaf, and we might then from time we might see it spread throughout the rest of the plant. It's colonizing. It's getting into other parts of that plant. It might even be moving into other plants that are nearby. Reproduction. Typically, those pathogens are going to reproduce, are going to multiply. With fungi and nematodes, it usually takes a few days to maybe even a few weeks in order for them to reproduce in large numbers. For bacteria and viruses, they can do that within a few minutes. Typically, bacteria can double their numbers every 20 minutes. So if you've got a bacterial problem, that's one that's going to really hit you hard and hit you fast. Fortunately, we've only got a few bacteria problems that we deal with plants. Most of them are fungal. But with fungal, it takes several days or weeks for those numbers to really build and multiply. And a lot of times with fungal, once we really see those signs and symptoms, we've got a large colony already established. And so is it too late? I don't know. All depends on the fungus. And then after reproduction, dissemination. They're going to spread. So the products of the pathogen reproduction are dispersed or spread to other plants. And that cycle continues, and it starts all over by depositing, deposition, penetration. So this is our disease cycle. I guess it would be better represented if it was drawn in a circle, because from dissemination it starts right back over with deposition. So we're going to focus on 4 types of plant pathogens. Bacteria. With our bacteria, they're single cells. And they generally enter the plant by way of a wound or a natural opening. Viruses are acellular. They really, as far as a virus goes, it's really not an organized cell. With bacteria and nematodes and fungi, they're considered to be living organisms. Viruses are really just strands of nucleic acid that are just kind of floating around there. They're really not a living creature, so they're considered acellular. But they're parasitic in that they rely on a host in order to reproduce. And many times these viruses are spread by insects. We're going to talk some about nematodes. Nematodes are microscopic roundworms, so they are actually an animal. And of the 4 pathogens, these are probably the most structurally complex because they're an animal as opposed to single-celled organisms. And then we've got fungi. Fungi they reproduce by spores. And of all of our plant pathogens, this one is probably the most common. This is the one that's going to cause the most issue. Now, yes, we're referring to these as pathogens. Pathogens, disease-causing organisms, but I want to say at this point that not all bacteria are bad, not all nematodes are bad, not all fungi, not all viruses are bad. There are beneficial ones out there. Which in 2 weeks, whenever we have our compost class, you'll learn that compost breaks down because of the activity of fungi. There are beneficial nematodes in the soil. There are beneficial bacteria. Unfortunately, it's the few that cause diseases that give them all a bad name. So realize that while we are referring to them as pathogens, there are good guys as well. They're not all bad. All right, that's just a quick overview about plant disease. Any question about that? If not, we're going to move on and go into a little bit more detail about each of those 4 pathogens. And then we're going to take a break. So, we've talked about, what makes a plant sick, and we've talked about the non-living versus the living or the biotic factors. So, with bacteria, with fungi, with nematodes, with viruses, we're going to consider those as biotic or those living type factors that make a plant sick. So we're going to start off with fungus or fungi. So fungi, they are living organisms with the same basic nutritional requirements for growth and reproduction as other living creatures. So they need moisture, they need nutrients, just like you and I need nutrients. Now they're not going to feed on steak and potatoes. Well, they might feed on potatoes, but they're not going to feed on steak. But they do have those same nutritional requirements as any other living creature. Many of our fungi we can see with the naked eye. If we see mushroom growing up in the yard, or if we see a shelf fungus on a tree, we can see those with the naked eye. Others you can only see with either maybe a hand lens or a microscope because they're so small. One of the tools that I have here in the office, whenever people sometimes bring in plants and say, hey, what's wrong with it? I keep a hand lens. So that if it is a fungal problem, I can look at it with that hand lens and get a better idea of what's going on. Another tool that I've got here is I've got a microscope that I can hook up to my computer and flash the image on the screen. And in some cases I have to use that microscope in order to see what's going on. And if I can't figure out what's going on, a lot of times I'll use that microscope to take pictures and I'll send it to the lab for diagnosis. So sometimes we can see the fungi, other times we can't. It's estimated that there's between 100,000 and 250,000 different species of fungi throughout the world. And that number's probably on the low side because as science is getting more and more advanced, we're finding more and more species of fungi. There's 2 different classifications or 2 different categories of fungi. There's what we call saprophytes. And these saprophytes, they grow on dead organic material. So if you go out and you see a fallen tree and you see some of that shelf fungi or you see mushrooms growing out of that, that's a saprophyte. It's feeding and breaking down that organic material. You see mushrooms growing in your yard, that's a saprophyte. They're feeding on some of that organic material that's breaking down in your yard. So that's one category of fungi. Then we've got parasites. The parasites, they overcome the host defense mechanisms through the production of enzymes, toxins, and other mechanisms. This is what affects your peaches. This is what gets onto the leaves. You've got perfectly good living tissue, and they're parasitizing or they're feeding off of that living tissue. So the saprophytes grow on dead tissue, dead organic sources, whereas the parasites feed on that live tissue. And most of the fungal diseases that we concern ourselves with focus on these parasites. When we look at the structure of fungus or fungi, they have a cell wall. And those cell walls contain living protoplasm. So we're talking about a living organism here. They're different from plants in that fungi do not have chlorophyll. Chlorophyll is that substance that allows plants to conduct photosynthesis. It gives it the green color, and through photosynthesis they take in the carbon dioxide and they release oxygen. Well, fungi does not have chlorophyll. Because they don't have chlorophyll, they have to obtain their energy from some other source. So that's when they're feeding on either that dead organic tissue or that live tissue. And so for fungal plant pathogens, that external source they obtain energy is the living plant. So that's the one. Those are the type of fungus that we're concerned about when we talk about plant diseases. So whenever we look at some, this picture was taken under a microscope. A spore producing. So we'll talk here in a little bit that fungi reproduce by spores. So spore producing hyphae. Hyphae are little arms or little structures in which the fungi will grow. So it's microscopic thread-like filaments called hyphae. So if you look at bread mold, Or if you look at a box of, in this case, a box of fruit that's been left out too long, and you start to see that fuzziness. That fuzziness that you're seeing is the hyphae. A mass of that hyphae is known as mycelium. So one hyphae is that strand or that microscopic filament. A whole bunch of that is known as mycelium. And so that's what you get whenever you get mold. That bread mold or that fuzzy mold is a mass of hyphae or mycelium. So with fungi, whenever you get to this point, it's pretty obvious or it's pretty easy to say, yeah, that's a fungus. I might not know what type of fungus it is, but I'll know it's fuzzy, it's a fungus. Bacteria isn't so easy to tell us what it is, but we can tell with fungus. But not all fungus, or you're not necessarily going to see a fuzzy mass on all fungal diseases. And as I've said, fungus cause more of our plant diseases than any other of our pathogens. Here's a picture of late blight on potatoes, and you can see this white discoloration. You can kind of see some of the hyphae growing around on the potato. It's cut open. You can see a little bit of that fuzziness there. Chestnut blight. This discolored area right here is where our fungal growth has started. We're not seeing those hyphae, but knowing if we were to really study about trees, and we would know, okay, this discoloration here, this is where it's starting from. This is my problem. And then Dutch elm disease. Dutch elm is also caused by a fungal issue. So fungus, they create more of our plant diseases than any of our other pathogens. So how do our fungal pathogens spread? They're spread by the wind, or they're disseminated by the wind. The wind blows and it blows those spores to other areas, to other plants. It's spread by rain. Whenever it rains, that rain falls on that plant tissue, that infected plant tissue. It can pick up those spores as that rain splatters or as it runs off. It transports those spores to other plants. Or it can be transmitted by insects. Insects are on the plants. They're feeding on the plants. They're on that plant, on that infected plant area. They're picking up some of those spores. They go feed on another plant. They're taking those spores with them. So rain, wind, insects, a lot of times we're responsible for fungal or pathogen movement in our own actions. Maybe we don't sanitize our tools. We're working, we've got a hoe or we've got a shovel or we've got pruners. We're done. We're going to go work on something else. Well, between maybe working here and working over there, if I'm working on some diseased plants over here, maybe I need to sanitize my tools just so I don't transmit that pathogen somewhere else. So a lot of times we're responsible for that transmission. Sometimes too, we've got our infected plants. Funguses are a good example of this. The leaves are infected, they fall off, or we let the leaves lay on the ground. A good practice to get in the habit of is to raking up that leaf material, especially with trees or even in your garden with some of those vegetables. Rake up those infected leaves and get them out of your garden. Because if you leave them there thinking, that leaf material is going to break down, I'm going to have organic matter for next year. Well, yeah, you're going to have that organic matter for next year, but you're also going to have that fungus still in the ground. And what's going to happen next year? You're going to plant your plants there, they're going to pick up that fungus, and you're going to have the same disease problems. So if you've got a diseased plant and it's too far gone, remove it. Remove the fallen leaves or even the fallen fruit that's on the ground. Otherwise, you're just going to keep that pathogen in your soil. So we, in many cases, are responsible for the dissemination of a lot of our plant diseases. With the different funguses, with the different fungi, a lot of times the way in which they reproduce is one of the best ways that we can properly identify that fungi. And many reproduce by forming a microscopic structure called a spore. If you've ever, I know as I've walked in my front yard, I've seen these little bitty white marshmallow looking puffs come out of the ground. It's a mushroom, it's a type of fungi. And whenever you step on them, or whenever I step on them, I see this black cloud of spores actually kind of drift. And as I've stepped on them, what I've done is I've released the spores. Those spores are the reproductive structures of fungi. I don't want to call it an egg because it's not an egg, but they're very light, they're very small, and they drift easily in the wind. And with many of our fungi, they don't require a male and a female like with flowers or with animals. A lot of times they can reproduce asexually, meaning they can do it all on their own. They don't need any other fungi in order to reproduce. Not all fungi look the same in regards to when they do reproduce. They can vary in size or color or shape. I mean, these are 3 different fungi right here. Here we've got penicillium on corn. We can see that little bit of fuzziness. I know, very similar to like a bread mold. Then we've got Helvella fungi, fruiting bodies. This is a type of mushroom. This one is breaking down organic matter. It's not the pretty white mushroom like what we're used to, or doesn't look like what the Smurfs lived under, but we've got a type of mushroom here. And then we've got these bird nest fungi. They're very small, they're not very large at all, but you'll see what looks like a little bitty cup, and if you look in it, you'll actually see these little black circular structures. Well, we do have these in this area, but they produce in different ways, so they don't all have the same type of size or color or shape, especially when it comes to reproduction and spore production. And they can produce millions of these spores at one time. And as I said, in some cases they reproduce asexually. They can produce their spores themselves. In some types of fungi, in order for those spores to be viable, it might require some sort of sexual reproduction. Whenever we see these shelf fungi on a tree, that's the fruiting body. That's the fungi that we're seeing. It's feeding on dead or decaying tissue. It's doing its job. It's breaking it down. But it's going to produce some sort of spore in order to help reproduce and to prolong its progeny, its offspring. So, fungi infect plants. What kind of economic damage do they cause? Well, they cause crop damage because they can directly infect or affect the crop, whether it be the seed, the kernel, the fruit, the nut, and that can affect the appearance, the taste, the nutritional value. That affects quality. And then it can indirectly affect the plant by reducing the load that the plant can produce. That affects quantity. So it can affect directly by affecting the quality. It can affect indirectly by causing the plant to produce less fruit. That produces, that's a reduction in quantity. And then of course, in order to treat and to control these fungal issues, you've got labor costs, you've got the purchase of fungicides, you've got the time that you're devoting in order to combat these funguses. So there is an economic impact in regards to fungal diseases. A few symptoms, if we're out in our garden, if we're out in our flower bed, if we're out and we see something that just doesn't look right, what are some of the symptoms we need to look for with fungi? Depending upon the fungi, it's either going to be localized, meaning that we're going to see it only a small part of the plant, or it might be systemic, meaning that the whole plant is affected. So with a leaf spot, we might see maybe a few dots or maybe a few spots on a leaf. With dampening off, maybe we're going to see some fungal growth and some damage along the stem, maybe down near the root. With rust issues, we're going to see an orangish or reddish growth along the blade. In this case, we've got corn rust, so this is along the leaves of the corn. We might even see a rust growth, maybe with some of our turf grasses, or I've seen this especially in moist conditions, downy mildew. It's that grayish, powdery type fungal growth that you can rub it off with your finger, and if you blow on it, you might even see some of the spores being disseminated or released. Those are some of the things to look for. Look at the leaves. Look at the structures. Feel the structures. As we diagnose a fungal disease, a lot of times you can see these fungal problems with the naked eye. You don't necessarily have to get a microscope or a hand lens in order to see a fungal issue. but you're going to have to look at the symptoms, and in order to maybe narrow it down. Okay, I know this is fungal, and in order to figure out maybe what type of fungal problem it is, you maybe have to do a little homework, do a little research. You might have to end up sending it to the lab. But we can diagnose fungal issues by looking at the patterns of symptoms of the entire plant population. Am I seeing this just on one plant, or am I seeing this on all of the plants in my garden? Am I seeing any of those fruiting bodies? Am I seeing any of the fuzziness of the mycelium? And so, looking at different things in the landscape, in the garden. So here are a few fungal problems that we might notice. Anthracnose of camellia. One of the things with fungal problems, with fungal leaf spots and spots that are affecting the leaves, with fungi you're going to see generally it's going to be rounded in shape. You can see we've got a round border, we've got a round margin here. It's going to be round, whereas with bacteria it's going to be more angular. It's going to be mainly contained within the veins. Fungi are going to be round. You're going to see that round discoloration or that round shape in the alteration of the leaf. Even here with I know this one's difficult to see, but we've got a leaf spot. We've got round dots. So that tells us I might not know exactly what's going on, but because it's round, there's no yellow halo effect on it. I'm probably dealing with something fungal. Even up here, as we look at these leaf spots in our hawthorn, they're round. So if it's round or got a round shape to it without a yellow halo, chances are it's fungal. We've got a leaf spot down here. These leaf spots are getting to the point where they're affecting the whole leaf. Here we've got a condition on a tree, butt rot. Well, as we look at the tree from a distance, well, it's orange or it's yellow. The leaves have turned, so maybe it's fall. I don't know. Maybe it's time for those leaves to turn. But as we look a little bit closer, and as we look at what's going on at the base of the tree, we see those shelf-type fungi growing out of the trunk of the tree. So something else is going on. It's not because it's fall that these leaves are turning colors. It's because we've got a fungal issue going down in the tree, this butt rot. So that tells me that I've either got a nutritional problem or this tree is on its way out. So what can I do to treat that? And I need to address it. I might need to address it pretty soon because it looks like this tree is right along the road. So I don't want this to fall and maybe damage my car or my truck or somebody else's car or truck. Some other symptoms or things to look for. Look for those visible fungal structures. Maybe you're going to see that mycelium or you're going to see those hyphae. Look for that fuzziness. You look for those round circular structures. Now I realize in this case we do have that yellow halo effect, but the discoloration is round as opposed to real angular. And then also, so look for that mycelium or just look for that fuzziness on the leaf. And those are some pretty good indications that you've got a fungal disease. But as I said, not all fungi are bad. We've got those saprophytes which degrade dead plants, and as they degrade those dead plants, that organic matter, they're releasing those nutrients back into the soil. That's a good thing. It's those saprophytes that help to make compost. It's those saprophytes that break down all of the organic matter that's in our forest and in our lawn clippings and return those nutrients back. And then also, whenever we plant legumes, it's those fungal relationships that legume establishes with the fungi in the soil that helps that plant to grow and to be able to release that nitrogen back into the soil. So not all fungi are bad. There are some good guys out there. There's more good guys than there are bad guys. Unfortunately, it's the bad guys that give all fungi a bad name. We're going to move on to bacteria. Bacteria are very primitive. They're single-celled organisms. Very primitive, especially when we compare them to fungi or nematodes. As I said, they're unicellular, one-celled, and some of them, like those spiroplasms and those mycoplasmas, They lack a cell wall. They don't have a cell wall. That picture there is several bacteria put together. You can see one there, one there, one there, one there, one there, one there. So you've got a cluster of bacteria, single-celled organisms. They reproduce asexually, or they can reproduce by binary fission. They can just, they are designed such that they can just split in two through a binary fission process. And whenever they do split by binary fission, you've got two separate but identical cells. And under ideal conditions, like I said, they can double their numbers every 20 minutes. So if you've got one bacteria, in 20 minutes you've got two, and another 20 minutes you've got four. I mean, you can do the math, and over the course of a day, you could have millions of bacteria cells. So because they can reproduce so quickly, it leads to a very rapid buildup of bacteria in plant cells. And bacteria can survive almost anywhere. We've got bacteria in the soil. Now let me go back and say, as with fungi, we've got good bacteria too, so not all bacteria are bad. But there is bacteria that lives in the soil. And it's that bacteria that can live in the soil that can maybe be taken up, maybe that can affect our plants next year. So we want to have good sanitation in our gardens and landscapes. We're going to have some bacteria. That bacteria can reside in plant residue. So that's why if we've got some sickly plants, we want to make sure that we collect all of that plant material and get rid of it, dispose of it. We don't want to continue to harbor that bacteria in our gardens or landscapes. And then also that bacteria can survive in living plants as well. So bacteria, pathogens are spread in many different ways. With fungi, we said it was dispersed by wind, by water, and by animals or insects. With bacteria, they can also be spread by water. So either by rain or by irrigation, they can be washed off and moved to other plants. We said that bacteria can live in the soil. So they've got movement of soil by wind. So as the wind blows away our soil through erosion, it can be taking bacteria along with it. Also by running water, so if water is flowing through, it can pick up that bacteria from the soil. Also our equipment, maybe our tillers or our hose or our shovels, can pick up some of that bacteria that might be in the soil and we can unknowingly transmit it to other parts of our garden. Infected seed. So sometimes unknowingly we go and we get seed, and usually when you buy seed from the store or from a reputable dealer, you don't have to worry about the bacteria. But maybe your neighbor or your friend gave you some seed in good faith. Here, have some seed and grow this, and unknowingly maybe they transmitted or they gave you some bacteria. I know earlier this year many people got the seed from China. This is an unopened package of seed from China. Yeah, it says it was a ring, but no, there's no ring in here. This is seed from China. I know that the Texas Department of Agriculture got many calls about it. I got several calls about it. The USDA got calls about this seed from China, and people were asked to send it in. In many cases, it came back as just seed. But the fear was, well, what is this seed? Is this going to be an invasive plant species that's going to take over? Or, even like in many cases it was something as simple as squash, were those seeds infected with some sort of bacteria, hoping for whoever sent this seed, those people hoping to release some sort of biosecurity issue? Know where your seed's coming from. Is it from a reputable source? Because we can transmit bacteria through infected seed. We can transmit bacteria through infected plant material from insects. Insects serve as vectors. We've talked about some of those cultural practices like contaminated tools, and then even through our own, unknowingly we walk through, and maybe as we're walking through our garden, we're walking through some of this infected plant material. Maybe we get some transfer onto our shoes. We're walking through another part of our garden. We've unknowingly taken some of that contaminated soil and transferred it. So it's always a good idea to, wash your shoes or keep your shoes clean. Even maybe rubber boots, and spray them off and disinfect them between uses, just so you're not transmitting potential pathogens to other parts of your garden. Mentioned also that bacteria can get into plants by microscopic wounds or by wounds in the plant. So we have heavy rain and wind, we have lightning, sometimes lightning might strike our tree or the wind blows a big branch down, we've got this big open wound. It's a great area for bacteria to be able to enter that tree. Insects as they feed on our plants can transmit bacteria, and then through some of our farming practices also we can potentially create microscopic wounds in those plants which can allow bacteria to come in. But then there's also some natural openings within our plants that we have no control over that allow bacteria to come in. So within the leaves of our plant, we have the stomata. The stomata is kind of what helps those leaves transpire. The carbon dioxide come in, the oxygen go out. That's a natural opening in the leaf. Bacteria can get into those. Water pores, those water pores help with the evapotranspiration of water in and out of the plant. And then we've got these lenticels. Lenticels are just microscopic or very small openings within the bark of a plant or within the stem of a plant. Naturally occurring. It's not as the result of a wound because of human intervention or anything. It's just naturally occurring. Bacteria can enter a plant through those as well. So we might do everything sanitation-wise, everything we can do to keep this at bay, but knowing we've got these natural openings as well. So there is a potential potential avenue for bacteria to enter our plants. So we talked about with fungi, some of the signs or symptoms to look for was those rounded discolorations or abnormalities on the leaves, looking for that fuzzy mycelium. What do we look for in bacteria? Well, with bacteria, you want some of the symptoms to look for and be slime. All of a sudden, I had flashbacks to Marvin Zindler. Slime in the ice machine. All right, for those of y'all who know who Marvin Zindler was. Water soaking, you're going to have like a depressed area that looks like a water-soaked area, or you might even have a foul odor. With fungi, you might have an odor. Generally, it's going to be a sweet odor, whereas with bacteria, it's going to be more of a foul odor. So leaf spots. If you look at the leaves, you're going to see, whereas with fungal we have those round spots, these are going to be more angular. They're not going to be round or circular. They're going to be more angular. You might see wilting. Well, is this a bacterial issue or is this something as a result of a lack of water? I don't know. But wilting is one of the signs of a bacteria problem. You're going to see a blight where parts of the leaves are going to turn brown. They might shrivel up a little bit. A blight, that causes the tissue of the plant parts to collapse. And then you might even see some rot. It's going to be soft, it's going to be mushy, you might get some of that water-soaked appearance. So those are some of the symptoms: leaf spots, wilting, blight, or soft rot. Those are some things to look for in trying to figure out, okay, what's going on? Okay, it's these. I think I might have a bacterial problem. Really, in order to know for sure that it's a bacterial problem and what type of bacteria it is, it's going to take a laboratory analysis. And of course, those cost money. Now, sometimes you can take a picture and send it to the lab, and they can say, "Oh yeah, that's this," or "We've narrowed it down, and it's this, or this." Now, if you do send a sample to the lab, there's 3 different types of tests that you could ask for. There is the biochemical test, which identifies fatty acid profiles. So some bacteria, they produce certain fatty acids that make their identification easy. Serological tests. They're looking at proteins and carbohydrates that are produced by those bacteria. And then there's the molecular genetic techniques, which actually look at the genes, look at the DNA of those bacteria in order to figure out what type of bacteria you're dealing with. So as we continue to talk a little bit about diagnosis, as we look at the leaves, we've got these blotches on the leaves. They're not round like they were with fungi. These are very angular, and in many cases they're limited by the veins. So here we've got some discoloration along the veins, or we might, if we had a large enough growth, it might be limited by these 2 leaf veins here. They're not going to cross over the veins like fungi would. So if it's angular and it's contained within the veins, you're probably dealing with a bacterial issue. Now, something else to look for, like, now wait a minute, now those look somewhat round. I thought you said that fungi was round. Well, they're round, but if you notice, it's got this yellow halo effect. Fungi will not show that yellow halo effect. When we see that yellow halo effect, that's a pretty good indication that we're dealing with a bacterial issue. That yellow halo that we're seeing that's part of the spread of that bacteria through the leaf. This is the tissue that's going to die next, or that's going to show symptoms next. So that yellow halo shows that bacteria actually moving through that leaf material. And as we look at it also, well, it's there along the vein. It's within the veins. So if you see that yellow halo, even if it does look circular, that yellow halo is a pretty good indication we're dealing with bacteria. Other signs or symptoms that you're dealing with a bacteria, if you cut the stem and you see this ooze or this stringy ooze oozing from the stem, it's a pretty good indication you're dealing with a bacterial issue. Or if you send it to the lab, they'll put it on a plate, then they'll culture it out, and you'll see the bacterial growth. And depending upon what test they do, that they can then determine what type of bacteria it is. For most of us, though, we're gonna look at it visually, or we might do this. We're not going to focus on that.
If you have bacteria, kiss it goodbye, or what?
That's going to be the easiest thing. Kiss it goodbye. Pull it up, throw it in your trash can. Do not put it in your compost bin.
Okay. Because I have a plant that I really like that.
Yeah. If it's bacterial, pull it out. And get rid of it. Yes.
Oh darn. Okay. Thanks.
So if we've got a bacterial disease, we can quarantine. By quarantining, especially if it's in a pot, it's easy to quarantine. We can pull it away. We can see if we can fix it by keeping it separated from the other plants. Cultural practices, prevent sanitizing our tools, cleaning our feet. There are some bactericides and antibiotics. Depending upon how far gone the plant is. Now, in your case, how far gone is the plant?
It's gone.
Okay, yeah. Pull it out. Get rid of it.
It's in a pot.
Okay.
And it's beautiful, and I loved it. And all of a sudden, it just died. And then it was a Hawaiian lay plant.
Okay.
A really different kind.
Mm-hmm.
And then it just started breaking off. And I saw that when you saw that stuff in it.
Yeah.
I'm thinking, what did I do? It was great. It was doing perfect. And
a lot of times with plant diseases, we've done nothing wrong. It's just nature.
I think it's the nursery I got it from, because that nursery is not doing well for a lot of plants.
Okay. So there are some bactericides you can use, all depending upon how far gone the plant is, as to whether you want to do that or not.
No.
There is some biological controls, and we're going to talk later on about some integrated pest management or some IPM techniques we can use, or getting those host resistance, getting those varieties that are resistant to certain types of bacteria. Those are your management options for bacteria. So yeah, I'm sorry to hear that about your plant.
I know. It was a weird, unique plant, but that's the way it is.
Yeah.
Plants are not people. They'll
true.
grow back.
They will grow back. You can always get more.
Always get more.
We're going to shift gears and move to viruses. Viruses are they're not considered living creatures like bacteria and fungi are. They're basically a protein coat that's made up of genetic material, either DNA, which is the double-stranded helix, or RNA, which is a single strand that contains genetic material in it. As I said, there's not necessarily any kind of cellular structure, and they can vary in shape. And these are so small, they can only be seen with an electron microscope. The microscope I've got under my desk is not going to work. It's going to take one of those electron microscopes in order to be able to see to see a virus, because they are so small. With a virus, it cannot make its own energy, so it relies on other hosts. So, and it is an obligate parasite that relies on host cells for survival. So obligate parasite means that it is relying on plants, it's relying on animals, it's relying on something else in order to survive, in order to reproduce. This is a tobacco plant that is affected by tobacco mosaic virus. Even tomatoes, there's a mosaic virus that affects tomatoes as well. Typically, when you get a virus, especially with a virus, that's when you just want to rip it out and get rid of it. Viruses can be transmitted by infected seed. They can also travel with pollen. It can infect pollen. We might unknowingly transmit a virus by taking an infected piece of plant material and grafting it on to a healthy plant. Or we've got, our insects that can certainly transmit viruses as well. So there's several different ways that we can transmit viruses. So a virus uses the host DNA in order to reproduce itself. So the genetic material that's in that virus is uploaded or works with the host DNA, and that's how it begins to replicate itself. And it can fill a cell with hundreds of thousands of copies of itself. So in a single cell, you could have hundreds, if not thousands, of copies of that virus within that single cell. And these viruses make proteins that trick the host into transporting those infectious cells to other parts of the cell, other parts of the plant, whether or plant or animal. So viruses can make these proteins that make the plant think, okay, this is normal, I'm going to take this to the leaf as it flows through the xylem. And unknowingly that plant has infected other parts because of the virus. Wide range of symptoms in regards to what viruses show. With a virus, you're going to see a reduction in quality, you're going to see a reduction in quantity. And with a virus, depending upon the virus, it might be localized, it might be limited to just one part of the plant, or it might be systemic, whereas it affects the whole plant. So as we look at some symptoms, here's an example of a leaf that's affected by a mosaic virus. We can kind of see the stained glass effect of the leaf. We can see where the parts of that leaf have been eaten away, or not necessarily eaten away, but the structure of the plant material has changed such that you can almost see through this leaf, giving it that mosaic type look. Another symptom is the stunting of the plant. We've got a healthy plant here in the middle. We've got two plants that are affected by viruses on either side. You can see that it's been stunted. We might see indication of a virus based upon the blotchiness of a fruit. We might see some discoloration, and we might think, well, maybe that's done by insect feeding or piercing on that fruit. Well, it could be, but if these insects can transmit viruses, maybe that insect fed on this, transmitted a virus, and that blotchiness is the result of a virus that was transmitted by an insect. Or we might also see some just basic modeling of the leaves. We might also see some genetic defects. Many times we might get a bag of pinto beans at the store. We might see some that are a little wonky or malformed. That could potentially be the result of a genetic effect. Could be caused by a virus, not necessarily saying that it is, but viruses do cause some genetic defects. We might have some change in growth habits. We might see necrosis or death on the fruit. We might see some yellowing or some spotting. Or we might see some chlorosis, or that yellowing of the leaves. Now this is when we have to scratch our head and think, okay, I'm seeing this chlorosis, I'm seeing this yellowing either of the leaf, or I'm seeing these spots. Is this a virus? Or is this maybe a nutritional issue? Is it maybe a lack of nitrogen? I don't know. So the easy thing to do would be, okay, I'm going to maybe give it a little bit of nitrogen and see if that fixes itself. If it doesn't, well, then maybe I could be dealing with a viral issue. So, whenever we start diagnosing and thinking through our plant problems, based upon other things that we've learned, we're going to have to use everything that's in our toolbox in order to help diagnose what our plant problems might be. So as we do begin to diagnose viral problems, check for symptoms previously shown. Have we seen this before? Have I seen this on other plants in my garden or in my orchard? Or is this something brand new? And eliminate some of those other potential causes. Is this insect damage? Is this a fertility issue? Is this caused by something else? Yes, no, okay, then maybe I'm dealing with a virus. And as I said, there's 3 tests for viruses, but you're going to have to send them to a lab. The electron microscope detects those virus particles. It looks at that genetic material. The serological test looks for proteins. And then the molecular biology, also looks for some of those nucleic acids or some of that genetic material. How do we manage for viruses? Well, unfortunately, there's only a few steps you can take for eliminating viral infections. One of them is to use virus-free seed, virus-free stock. Keep the equipment clean. Sanitize your hose, your rakes, the tools that you use. Rotate crops. So if you've got several different beds, and Tom Leroy talked about this when he talked about vegetables, with rotating crops, especially with your nightshades, your tomatoes, your potatoes, your peppers. Don't plant them in the same bed or in the same spot year after year after year, because you're going to end up with disease problems. So rotate them through different areas of the garden, and maybe over a 3-year period, they're going to rotate through different areas of the garden, so they'll come back to that spot maybe 3 years later. That will, by rotating the crops, you're going to reduce the instance of viral diseases and just diseases in general. We talked about removing any infected plants or plant parts, and if you need to, there are some viricides or pesticides that you can use for the control of viruses, or for the control of those vectors, those insects. So it might be that you're not necessarily applying a product to control viruses more so than you're applying an insecticide to control an insect that's going to potentially transmit a virus. Planting, you can plant those resistant varieties. Some plants have been bred specifically to resist certain viruses. I know, like tomatoes are a good example. You can look on that label and that series of letters. It'll tell you what diseases or what viruses those plants are resistant to. Becoming a little bit more common within the nursery industry to vaccinate plants. Plants might be infected with a mild strain of the virus, and that leads to resistance of a more harmful strain. So just like we're vaccinated for certain diseases, plants can also be vaccinated. You probably, it's done, but you're not going to see it on the label saying this plant has been vaccinated against viruses. You're not going to see that, but know that is a practice that is done within the nursery industry. And then heat treatment. Some viruses can be eliminated by infected plants by using heat treatment on the vegetative planting stock. So early in the process, they might heat the seed, or they might heat some of that vegetative material that they're using to grow those plants in order to kill any potential viruses that might be there. All right, we're going to do nematodes, and we're going to take a break. Nematodes are probably our most complex plant pathogen. They're complex in that they are an actual animal. They are microscopic roundworms. They have tissues, they have organs. Unlike bacteria or even fungi, which are anywhere from single cells to a few cells, these nematodes are multicellular. They've got a complete body system with a reproductive system and a digestive system. They have tissues and organs. Not all nematodes are bad. There are good beneficial nematodes, but then there are those pathogenic nematodes as well. Many nematodes have a unique feature that distinguishes how they feed. This lance nematode, it has a little stylet or a little spear that extends from the mouth, and that's how it feeds. This ring nematode, here we can see it's got an esophagus, and it's also got this little spear that comes out. Here we've got this carrot cyst nematode. I know it's hard to see in the picture, but that's its intestine. So with these microscopic roundworms, they actually have organ systems that allow it to feed and to grow. Nematodes are more like insects than they are fungi or bacteria. And nematodes can inhibit or they can live in the soil, they can live in the stems, they can live in the leaves, they can live in the roots. Probably the type of nematode that we're most familiar with is the root knot nematode. They feed on the roots, and as a result of them feeding on the roots, and I've got a picture here in a little bit, but as a result of them feeding on the roots, they develop nodules on the roots that inhibit the uptake of nutrients from the soil, and it causes the plant to wilt and die. There are thousands of species of nematodes, but only a few of those thousands of species are of concern for plants as far as parasites go. They require water in the soil, so they require soil moisture in order to move in the soil. And you're thinking, well, if I just alter the environment, maybe I can control the movement of nematodes and I'm not going to have to worry about that. Well, the problem with root knot nematodes, as well as all nematodes, they require the same environment that the plant needs in order to survive. So you can't just cut back on the water, you can't just cut back on one of those environmental factors in order to control nematodes.
Question. Tom Leroy was talking about planting a certain kind of ryegrass that kills nematodes.
Elbon cereal rye.
Yeah, and it has ergot in it, correct?
I believe that's correct, yes.
Okay, I was just wondering, because isn't ergot what caused, they think caused the Salem witch trials?
Yes.
I'm just wondering if it's all related. Sorry about that.
Yeah, no, and that's a good question. I don't know. But yeah, like Tom said, with that Elbon cereal rye, they're not exactly sure what attracts the nematodes to that Elbon cereal rye, but that Elbon cereal rye, those nematodes get trapped up in the root system of that. So that's one method that works for the control of those nematodes.
Sorry about that.
No, good question. And we're here in Texas, because of our warm climates, we're in perfect climate for nematodes. They especially like a coarse textured soil. What type of soil do we have here in Leon County? A coarse, sandy textured soil. So we've got perfect environment for nematodes. But like I said, they're not all bad. It's just a few that give them a bad name. So with nematodes, they actually grow from eggs. They go through 4 juvenile stages until they reach adulthood. Some nematode species they evolve through asexual reproduction. Many of them are sexual, meaning they need both male and female. And depending upon the host will depend upon the level of reproduction for nematodes. So in this case here, yellow potato cyst nematode eggs emerging from the cyst. So the female will lay a cyst, or like an egg, and those little nematodes can come out of there. Here's a blown up picture of those little nematodes in their egg casing. And then here's one that's hatching out of his egg casing. So many of our nematodes require a certain host. So all plant parasitic nematodes are obligate parasites. They require that host in order to get their nutrients, in order to be able to reproduce. They have to have that certain host. Some have a broad range of hosts where it's a plant, if it's a certain type of plant, we can live on it. Others need a specific type of host in order to reproduce. So many times with nematodes, we don't know what the problem is because we don't see the signs or symptoms like we do with fungi or bacteria or even viruses. In this case, we've got a pine tree that's died. Well, is this drought related? Actually, in this case, we've got these pine wilt nematodes that feeds on the xylem and phloem of the pine tree. So these nematodes are in the tree and they've affected the flow of nutrients up and down the tree, so it's killed the tree that way. We've got these stubby roots on these here, and we've got nematodes that have actually eaten some of the meristem or eaten some of the root material, causing the stunting of those plants there. And here we've got nematodes feeding on the surface of a leaf. So they don't have to be feeding on roots. You know, they can be in the trunk, or they can be feeding on the leaf as well. Typically, I'll get to that point about what the nematodes we have here. But you know, these nematodes, the pathogenic nematodes are parasites. So they're going to feed off that host. So we've got migratory ectoparasites. These ectoparasites stay on the outside of the plant, and they feed on plant surfaces, and they remain mobile throughout their lives. So as that water flows through the soil, those nematodes are going to travel through that soil water. So they remain mobile. Whereas we also have migratory endoparasites, and these endoparasites feed on the internal plant tissue. So they get inside the plant, and they remain mobile through the plant for their entire lives. Here's an example of what a root knot nematode can do. These sedentary parasites, they can either be endoparasites, meaning they get in, or semi-endoparasitic, where their bodies might be inside the root, or they might be on the root surface at maturity. They can kind of go back and forth. But once they begin feeding, they begin to lose that mobility. So with some of our root knot nematodes, they're an example of one of those sedentary parasites. And we can see some of the damage caused by the roots, or caused to the roots by those root knot nematodes. In many cases, especially with our root knot nematodes, and that's probably the main nematode pathogen that we have here in this area. You know, the plant's doing fine, and all of a sudden it begins to show some wilt and distress, everything. But you don't know what's going on, and if you dig down, you can actually and you uncover a little bit of the root. And you see those knots or those nodules. That's how you can figure out that I've probably got nematode problems. So some of the signs and symptoms above the ground, you might see some chlorosis or some general stunting. Well, wait a minute. Chlorosis and stunting, those were also some of the symptoms of viruses. So am I dealing with a virus, or am I dealing with a nematode? Well, that's when you have to take all of your knowledge and start putting two and two together and seeing what you come up with. Nematode symptoms also, you're going to see affected plants usually distributed in irregular or patchy patterns. You might have one over here, you might have one over here, whereas with a virus, you're probably going to have all of the plants in the same area affected, whereas with nematodes, they might be kind of scattered. Plant death occurs only in most severe cases, so with nematodes, it's not necessarily going to kill the plant unless it's a severe case. Many times, it's mistaken for soil fertility problems. So in order to diagnose nematodes, dig down, look at those roots. Do you see those big growth nodules on the roots? It might be that you have to send that plant sample or that root sample to a lab. Now, like I said, with root knot nematodes, you can see those galls or those growths on the root. So that's a pretty easy way to determine if you've got root knot nematodes. So what can we do to help eliminate or reduce our chance of nematodes? Crop rotation. There are varieties that are resistant to nematodes. We can treat the soil with a nematicide. Basically, that's a fumigant or a chemical to kill nematodes in the soil. There are some non-chemical or some non-fumigant methods. They're not necessarily as effective, but they do provide a little bit of control. So as I said, very small roundworms, typically in the soil. They'll attack roots, sometimes the foliage, sometimes they get in the trunk, and they reproduce by eggs. So that's nematodes. Now I mentioned earlier about parasitic plants. I want to finish this section by talking about parasitic plants, and we'll take a quick break. Parasitic plants, mistletoe and dodder, obtain all or some of their nutrients from other plants. They may or may not have chlorophyll, and when compared to some of our other disease pathogens, parasitic plants really don't cause that much problem. Yeah, they feed off other plants, but now we've got the mistletoe. I know sometimes in the wintertime, as I look up at my trees, and I'm thinking, well, golly geez, at least I've got some green. it's mistletoe. I've got some green. I don't like it, and I'm not climbing up there to get it out. But I do have to realize that this mistletoe is taking nutrients from my trees in order to grow. And then the other one that I mentioned is dodder. Dodder is very viney, very stringy. I'm not aware of any cases of dodder here in Leon County. There could be, but I'm not aware of any. But dodder is a parasitic plant found in other parts of the state. In many cases, these are transported by maybe either birds or by the wind. Now, I will say for mistletoe, if you do have mistletoe in a tree and it's at a place where you can get to it and control it, the best way to control mistletoe is to, wherever you see that mistletoe growing out of the branch, come back towards the trunk about 8 to 12 inches, and then you can cut there. Because what happens with mistletoe, as it takes root in the tree, it will send out feeder roots within that branch. And typically they're 8 to 12 inches long. So by cutting back about 12 inches towards the trunk, and then cutting it at that point, that will, I mean, yeah, you're going to eliminate that branch, but that will also get rid of the mistletoe in that tree. Unless you want it for Christmas, then you just go break off a little twig of mistletoe, put it above your door, and see who gives you a kiss on the way in. Now, regardless of what we're treating, whether it be fungus or bacteria or viruses or nematodes or even mistletoe, if you're using a fungicide or an insecticide, make sure that you read the label before you use the product. Read the label, know what you're using, know how to use it. Know if you're supposed to wear any kind of gloves or any special protection. And remember also that the label is the law. So whatever is written on that product label, that's how that product is to be used. And if you misuse that product in any kind of way, and it creates adverse effect on your neighbor's property, or if it does something, you could get in trouble for that. And it all goes back to, well, did you follow the directions on the label? So do realize that, I know this is hard to read. It's small. I mean, I need my glasses. I need a magnifying glass. I might need to pull out the microscope in order to read that. It's so small. But, you are, as the user, you are responsible for knowing and following the directions on that label. So, regardless of what you're using, a fungicide, an insecticide, whatever, know how to use it. So, with this, what kind of wraps up, these living or these biotic methods of disease and disease transmission. All right, well, let's go ahead and pick up. I don't have much left on plant disease, and then we can get to making jam.
Yeah.
So we're going to talk about some of the plant disease control principles. What are some things we can do to control disease? And then if we're going to send a sample to the lab, what do we do? So whenever we talk about controlling diseases, so there's 6 different things we can do: resistance, exclusion, protection, eradication, avoidance, and therapy. And we'll talk about each of these. Now with therapy, this isn't like Dr. Phil and come on, let's can we talk? Nothing like that. But we're going to talk about each of those. So when we talk about resistance, that's planting those varieties that are naturally resistant to the pathogen. I know I mentioned tomatoes earlier. You can look at in many of the seed packets, they still have these letters listed. V meant that it was resistant to verticillium wilt. F meant that it was resistant to fusarium wilt. N was for nematodes. A whole alphabet full of letters. So if in those seed packets, many of them still have those. If it is a resistant variety, it does list what it is resistant to. So you can find that information on seed labels and seed catalogs. The internet might have some stuff on that. Research publications. But you want to make sure that you are careful of the reliability of the source. Make sure that you are looking, especially if you're doing this internet research about resistant varieties, make sure that you're looking at a reliable source. Another disease control technique is exclusion, basically keeping those pathogens out, using only healthy materials, making sure your equipment is clean,
What do you clean with? I'm sorry to interrupt.
You can use just like a bleach solution.
Okay.
You know, mix up a bleach solution and spray that on your shovels, on your hose.
Okay.
The thing is with exclusion is, you know, you can't necessarily control everything. But that's certainly a tool that you can add to your toolbox. Protection, you can utilize some sort of barrier between the plant and the pathogen. This might be a chemical barrier such as maybe some sort of fungicide or maybe an insecticide. It might be some sort of biological barrier, maybe in companion plants. There's research being done about, you know, if I plant this type of plant maybe as a border, it's going to attract the insects here and maybe leave my more prized plants alone. So maybe by planting some sort of biological barrier or even a physical barrier, if we can put a cage around our plants, maybe we can keep out insects or birds or those deer that are going to eat on our plants. So maybe those are some different ways. And then of course, biological, who could forget, you know, of course, using those beneficial insects to help control aphids, mites, and other insect populations. Maybe we can eradicate, maybe we can kill or get rid of that pathogen. We've talked about this already, about, you know, raking up or getting rid of those leaves that fall underneath the plants that might be diseased. So getting rid of that plant debris. One thing we don't think a lot of is actually fumigating the area that we're going to be planting in. Now, whenever we talk about fumigating, we immediately think of chemicals. But it doesn't have to be chemical. We can solarize the soil. So if we've got an area that's maybe, we know it's got a disease problem, we can take clear plastic, not black plastic, but the clear plastic, put it over that area and kind of put some rocks or some soil around the edges. By putting that rock or soil around the edges, before you put the plastic down also, you want to wet it. Put that plastic on, seal the edges, and you want to do this typically probably June, July, August in the heat of the summer. That sun will bake this soil. The moisture in there will heat up, and you're going to create a very hot environment down here that's going to kill pathogens that are in the soil. It's also going to kill the good guys that are in the soil as well. So whenever you take that black plastic or that clear plastic off, you're probably going to have to do some soil amendment, maybe some organic matter or maybe some fertilizer to help restart that biology in the soil. But soil solarization is a great way, an easy way, in order to help get rid of some pathogens. Avoidance, we talked about crop rotation. By simply moving where you plant your crops from year to year to year, you can help avoid the instance of plant pathogens. And then therapy. Maybe you might use some chemicals in order to reduce or eliminate some of the pathogen problems. It might even require pruning, maybe pruning out an infected branch or an infected limb and getting rid of it. But by pruning, that's considered a therapy way of reducing and eliminating disease. Now, I used the word earlier, Integrated Pest Management, or IPM. IPM is a way of dealing with plant problems, plant diseases, where it doesn't necessarily involve chemical usage, but chemical usage is certainly one of the steps in an IPM process. So as with this triangle here, maybe we're going to start off by using non-chemical measures in order to treat for this pathogen. So maybe we might put out some light traps or some sticky traps, maybe to collect or to catch some of these insects that might be transmitting disease. It's not a chemical, but it's a sticky trap. We're collecting these insects. Or maybe we're putting out a pheromone trap. Pheromones, or insects are attracted to the pheromones because they're thinking they're about to breed. They're gonna be able to reproduce, and so they're attracted to these pheromone traps. They get in, they can't get out. So you're eliminating some of those insect populations. Maybe you're practicing crop rotation, or maybe you're using like a BT. BT, that Bacillus thuringiensis, which is that bacteria, that good bacteria that affects caterpillars. Maybe you're using something such as BT, or maybe you're using ladybugs, or maybe some of the parasitic wasps. Maybe you're utilizing that. That's a non-chemical control measure as well. Well, maybe you've done all that and you've still got problems. Well, that's where monitoring comes into effect. You know, having those feet on the ground and walking around. What are you seeing? Are you seeing insects? Are you seeing birds? Are you seeing wildlife damage? Or are you seeing signs of disease? What are you seeing? And then, after you've employed all those non-chemical measures, and you have a friend, he walks through his garden, and one of his non-chemical measures, he's got an old dust buster vacuum cleaner, cordless dust buster, and as he walks through the garden, as he sees those insects, he sucks them up in his dust buster. It's a non-chemical measure of controlling insects and disease. After you've done all of this other stuff, then, you know, that's when you might have to pull out the big guns and you might have to actually employ some of those chemical uses. But, you know, don't think that, oh, I've got a problem, I've immediately got to go to the chemical route in order to fix it. No, there's other things you can do first before you have to go for that chemical route. And as I've already said, if you use chemicals, know what you're using, read and follow the directions on the label, because you are responsible for what happens. Now, to kind of wrap up, we're going to talk about how to go about diagnosing. You know, kind of put some of those, give you some more of those tools in order to be able to maybe not identify specifically what you're dealing with. You know, okay, I know this is a bacteria. I don't know what kind. You know, maybe give you some of those tools where you can at least narrow it down. I usually start with this joke, and I forgot. So, you know, why did the guy bring a mushroom to the party? The mushroom, he's a fungi.
Fungi.
All right.
It's
All right. It was bad, but I'm sorry. I'll edit that one out. So, in order to do diagnostics, you know, we're going to take an organized, logical approach, and we need to learn to ask the right questions. Well, how do we know what the right questions are? We're going to talk about that. We might be utilizing reference materials. Some of those reference materials might be what's in your Master Gardener book. It might be some of the handouts that you've given. It's going to be talking to other people that know about plant disease. Going to be a little bit of knowledge about the weather conditions and the soil conditions. Has it been dry? Has it been rainy? And then just a little bit of common sense and, you know, a little bit of detective work. And each of you are capable of doing all of these. So with these simple tools right here, you can be a detective in order to help diagnose plant problems. So I think this graphic is in your handout. You know, five steps in field diagnostics when it comes to plant diseases. First off, be able to identify the plant. Now, you don't have to know specifically like, oh, this is a certain variety of salvia, just so long as you know it's a salvia. You know, that's the main thing. Be able to identify the plant. And then assess the nature of the problem. Is there really a problem? Or is this a natural part of the plant's life cycle? You know, the plant might be losing its leaves. Well, is it a problem? Well, we're getting into fall. During the fall, plants normally lose their leaves, so maybe it's not a problem. But if the plant's losing its leaves and it's middle of spring, we might have a problem. The third step in diagnostics, come up with a list of suspects. Is it fungal? Is it bacteria? Is it weather related? Is it, you know, what are our list of suspects? Who could the guilty parties be? Then we're going to narrow that list of suspects. Well, based upon some of the questions I've asked, some of the information, some of what I'm seeing, I can eliminate this, and this. So I'm narrowing my list of suspects. And then that fifth and final step in diagnostics is to get help as needed. And that help might be maybe bringing it to me here at the office, maybe talking to other master gardeners, maybe taking it to a nursery. You might have to send it to a diagnostic lab in order to get that. But those are the 5 steps in field diagnostics. So let's talk about each of those steps a little bit more in detail. So identify the plant. You know, as master gardeners, people are going to find out, hey, you're a master gardener, what's wrong with my plant? So, you know, let's find out, okay, what is the plant? Ask the person that, you know, that you're at their house or they've submitted the sample. What is this? And if you don't know, you know, maybe come to me, other master gardeners, look at books, you know. And if you can at least get it down to the genus. Like I said, you don't have to know what type of salvia it is. As long as you know it's a salvia or as long as you know it's an oak tree. You don't necessarily have to know what type of oak for the majority of our plant pathogens, just so long as you know it's an oak. So identify the plant and then define the problem. Is there a problem? Is this something that could be pathogen related, or is this something that could be normal? Or is this something that could be non-biotic, as a result of the environmental conditions? If it is a problem, could it be fungal? Is it be insect? What could the problem be? So, pop quiz. Somebody shows you this leaf. Is that a problem?
Yes. Yes, sir.
I don't know. I'm thinking it could be because it's round and it's there. And it looks like spores, but I'm not sure.
It's on the vein.
I will tell you that it is a little difficult with this picture, looking at it as a picture instead of if you had the actual leaf. This is actually not a problem. These are actual galls formed by gall wasp. They will deposit their eggs on the leaf of a plant, and it will encapsulate the little larvae. And these gall wasps are actually parasitic wasps that will feed on other insects. So it doesn't look good. I've been called out to look at trees, like, oh, my tree's got this bad disease, I don't know what it is. And it was covered with these little galls from these gall wasps. It doesn't look good, it's not going to hurt the tree. It's actually a good thing, because you've got these little parasitic wasps that are going to be feeding on other insects. But what about this one?
Yeah.
Yes.
Is there a problem?
Yes.
Yes.
Yeah, there is a problem. We know it's a problem. We've got this discoloration on the leaves. With a little investigation, a little bit closer, well, a better picture for one thing, but a little bit more research, we can maybe figure out, is this fungal or is it bacterial? But in this case, there is a problem. So just because it's not necessarily a problem, this is normal.
The leaf looks healthy there too.
The leaf was very healthy. Yes. Whereas with here, you can tell that there's some things going on. This just doesn't look right.
Not happy.
So as we define the problem, here's some of those questions to ask. What's the history of the planting and of the site? Have you always planted this vegetable in this spot every year for decades, or have you done some rotational grazing? What used to be on this site before you planted your plants here? And check for patterns. Is it just one plant that's showing these symptoms? Or is it the whole row? Or is it the whole flower bed that's exhibiting these symptoms? Or is it all of the plants in the community? Not just this one type of plant, but is it all the other plants in the community as well exhibiting these signs and symptoms? And if is it localized? Is it just one part of the plant, or is it affecting the whole plant? So can you identify the affected plant parts? Is it just the leaves that's affected? Is it the trunk, or is it the stem? What parts are being affected? So based upon those questions that you've asked and the answers that you've gotten, you can start to develop a suspect list. Well, what would cause the symptoms that I'm seeing? So if I'm seeing a leaf scorch, well, that could be caused by drought, that could be caused by root damage, that could be caused by bacteria, that could be caused by a root rot fungus. So I'm starting to develop my suspect list here. And as we develop our suspect list, we've got to remember, we've talked a lot about those biotic pathogens. Usually with the biotic, those symptoms are usually scattered. They develop gradually over time, and the sign of the pathogen is sometimes observed. Whereas we've got those abiotic factors, that's our environmental factors, whether it be our soil condition, whether it be too much, not enough water, maybe our air quality, our temperature. Usually those symptoms are uniform. You're going to see the same symptom over many plants within that community. And it's generally all going to appear at one time. Whereas with our plant pathogens, we're going to see it start here, and then over time we're going to see it spread, and it might spread to the next one over time. With our abiotic, or with our environmental conditions, generally all going to appear at once. And it doesn't appear to spread. I mean, it's all going to happen at once, and it's going to affect maybe all of the plants in the community. It might just affect one plant, and it's not going to spread. And it can affect more than one type of plant in that immediate area. So these are those non-living or those abiotic factors that's going to create, that give us these symptoms to help. So as we try to develop our suspect list, is it our bacteria, fungi, and viruses, or is it something else? So we've developed our suspect list. We're going to refine our diagnosis, or we're going to narrow down our suspect list by asking those right questions and matching those symptoms to the potential suspects. So scorching, maybe it's a lack of water to the leaves. Well, what prevents transport of water up into the plant, into the leaves? So using some of that knowledge that you've gained. And then also, in addition to thinking about those living or those biotic pathogens, what about those environmental conditions? Well, black spot on roses. Well, was there sufficient moisture? Has it been humid? Has it been rainy to be able to have black spot on roses? If it's been rainy conditions, that could be black spot on my roses. So matching up those environmental conditions with what the pathogen needs to grow will help you out. And then so matching up those notes taken and comparing it with what you found in your research. And if you have to get additional help, at Texas A&M there is the Texas Plant Disease Diagnostic Lab, a great source of information. There is a charge if you send them a sample. There is a charge for them to run their tests. Bring it to me a lot of times, where I can take pictures and we can send it to the diagnostic lab if I don't know what it is. And they can diagnose based on email pictures for no charge at all. But as you're getting that additional help, try and get as much information as possible. Know the history of the site. Know what the weather has been. Know if it's just the one plant or the several plants. Send the lab as much information as possible. Now in your handout, I have provided this flow chart. And this flow chart will help you diagnose plant problems as to whether is it a problem or is it just something naturally occurring. So I'm not going to go through this whole chart, but do realize that this is a resource I've provided you, and you can utilize this in order to help figure out what might be going on with your plants.
Can I get that one up?
It's in here.
Yeah, there's a big, look further in your handout, in the one with the glossary.
On page 23.
You look on the packet in front of you. If you look in that
23.
glossary packet,
Okay.
there should be, double check and make sure I put it in there in that
It's
glossary
here.
packet.
It's here.
Okay.
But yeah, there should be a big version of that blown up for you so you can read it. Actually,
Oh, okay. I'll look there. Thank
Yeah.
you.
If you do have to end up using the Texas Plant Disease Diagnostic Lab, this is their website. You just do a search for Texas Plant Disease Diagnostic Lab, and you can download the form that you would need in order to send a sample to them. There's instructions on how to prepare the sample, how much the test costs. A lot of great information. Even if you're not sending a sample, a lot of great information on that Texas Plant Disease Diagnostic Lab website. So if you're thinking about sending a sample, a few things you want to consider. You want to make sure that don't just clip off the infected area and send that off. If you can, send enough of the sample where you show the healthy and the infected area as well. You want to, if the lab can see that transition, they can get a better idea of what's going on with your sample. If possible, the entire plant. Now I realize it's difficult to pull up some plants and send them, especially like trees and big plants. But the thing is, if you can keep the sample fresh, it might be putting a little bit of ice pack in there. They're located in College Station, so if you're going to College Station to go shopping, make a plan and take it. You can drop it off at the lab. There's a form. There's a form on this form here, and it's going to ask several questions. Make sure the form is completed as full, as complete as possible. Times and dates. When did you first notice the symptoms? Have you done any type of chemical control recently? Could this be herbicide damage? And then also, in addition to sending in the plant sample, if you can send in some pictures of the plant, and not just the whole plant itself, but maybe some close-up pictures of the plant and the environment, the area that it's in, that will help piece some of those puzzle pieces together. So if you do send in a sample,
send the information form put it in a Ziploc bag and secure it tightly. That way it doesn't, because whenever you send in these plant samples, there might be some moisture or humidity condensation buildup. We don't want to damage this so that they can't read it. They need to be able to read that so they know what's going on and know how to get back with you with the results. So put that in, fill out that form, put it in a Ziploc bag, and place your sample in one Ziploc bag, place the completed form and any type of payment that might be needed in that separate Ziploc bag. And you'll send it to the lab. Now, if I'm going to send a sample to the lab, like I said, I want to have a sample where, if possible, I can show the diseased area as well as the non-diseased area. So I'm showing that transition zone. Now, in this case here with my crepe myrtle, I can't send the whole crepe myrtle. So maybe I'm going to prune off a branch, or maybe I can prune off a little twig. But I want to make, if I prune off a little twig, hopefully I can send more than just one twig. I want to be able to send several twigs so they can get a good representation or a good idea of what's going on. So in this case, the lab got this and said, hey, what's the white stuff? Well, chances are it's probably fungal, but what type of fungus is it? Another sample, this sample was submitted for oak wilt and bacterial leaf scorch. So you can see the affected leaves. You can see they even sent in some of the wood. Some of the they couldn't send in the whole tree, but they sent in a branch and some of the wood. With this palm, they sent in several of the palm fronds in order to figure out what was going on. Now, this is a bad sample. And because it's a bad sample, because, what's going on? Well, I see a twig with a few dead leaves on it. I don't have that transition zone. I don't know what the healthy stuff looked like. And in this case, the plant is so far gone, I don't know what it is or what's going on. And the same with this. You've got the leaves that are so far gone, there's no transition zone. There's no healthy material so I can compare it to. I also mentioned, in addition to sending samples, take a few pictures. And here's a few examples of what good pictures might include. I can see a picture of the whole tree. I can see the healthy part. I can see the unhealthy part. I can see what's going on. And in this case, they've given some information. They've given an address, the date when the picture was taken. That on the southwest corner there's poor drainage. On the southeast corner there's better drainage. And also it says that this is a Mexican sycamore. So so in this case we're looking at the trunk of the tree. Get some close-up pictures if you can. Here we've got pictures of the tree, several trees in a line. We can see healthy trees. We can see some of the symptoms that the trees are exhibiting otherwise. Now, as you take the photos, look at them before you send them. I've had people email me pictures like this one here. What's the problem? What's going on? What in the world am I looking at? For one thing, I think what their focus is out of focus. So I can't even make out what it is. Here, I'm guessing it's some sort of bacterial issue, just because the damage is between the veins and it's very angular. I'm seeing that yellow halo, but it's out of focus. And here, I think I'm looking at some roots, but I'm not exactly sure what I'm looking at. So take a look at the picture before you send it, and make sure that you can make out what the person that's going to be doing the diagnosis needs to be seeing. Now, a few resources. These are some internet resources. The plant clinic at A&M, the Texas Plant Disease Diagnostic Lab, plantclinic.tamu.edu. You can get those submittal forms there, information on how to package and send off a plant sample. The Plant Disease Handbook web resource that is actually maintained by the Plant Diagnostic Lab. You can log on to that site, and it breaks it down by trees and shrubs and vegetables and flowering plants. You can pull up, go to whatever type of plant you've got problems with, and there's a long listing of diseases that you might encounter.
Oh, wow.
So this is a good tool to use for diagnosing plant diseases. On the AgriLife bookstore site, there are several resources, many of them free and downloadable in regards to plant disease. And then also on YouTube, the Plant Disease Diagnostic Lab has a YouTube channel where on a weekly basis they put up an educational video. They call it What Wednesday. So every Wednesday they put up a little 1-minute video clip about something that they're seeing or that's going on with plant diseases across the state. Like this one is on. This one is on well, I can't read any of that, but there's stuff on insects or stuff on plant diseases. Actually, this one is on. Well, we'll just watch this one.
Howdy, fans, and happy Wednesday. Today, we get to ask Dr. David Apple, tree pathologist, about the significance of finding Bacterial fungi on trees.
So the fungus in the image is actually what we call wood-rotting Bacterial. That's because it's in the group of fungi referred to as the Bacterial. These organisms, for the most part, have the ability to live on dead, decaying organic matter, but a select few of them can actually kill a living tree. So we need to take that in consideration when we see it, because if the activity is too much for the tree, then it's going to be destabilized and it's going to blow down under high winds.
If you find this fungi on your tree, please have it checked out for structural integrity to avoid it becoming a hazard tree. See you next week.
Every Wednesday they put up a new little video. I mean, that's not very long, about a minute. And they've got their library on there, so you can go back, and they've got videos on there about how to send samples to the lab. They've got videos on there about the Chinese seed. So anyway, that's another resource for you as well. So that kind of concludes our presentation on plant pathology. Any questions? I know I was kind of a shotgun approach. We covered a lot. We didn't go into a lot of detail because I didn't want to go into too much detail because I don't want your head spinning. If you can tell the difference between a fungi and a bacteria, you've succeeded. You kind of know how to treat. Now, not all funguses are treated the same way. Not all bacteria are treated the same way. But at least if you can narrow down what pathogen you're dealing with, you're going to be leaps ahead about how to get a handle of it and how to control it.